I'm Kathleen Woulfe. I'm assistant professor in cardiology, and my research focuses on looking at age and sex-specific differences in the contractile unit of the heart, specifically the sarcomeric proteins. And today I'm going to tell you a little bit about our work looking at a post-translational modification and its functional consequences, and how they're different on different sarcomeric proteins. And so, as we all know, the body changes pretty significantly with aging. But one of the things that happens to the heart as you age is you have a prolonged or delayed relaxation. And this has been found with normal or healthy aging. And while we think about the heart as a pump and really focus on the contraction and squeezing blood out of the ventricles, we also know that relaxation is just as important. We need the heart to relax to its fullest to be able to fill up with an adequate amount of blood to be able to pump back out to the body. And conditions where we don't have that complete relaxation lead to heart failure as much as conditions where we don't have that contractile function. So my lab really focuses on the mechanisms of relaxation at the level of the sarcomere. So this picture is just again to emphasize that as we age, many things are happening in the heart. We have an increase in inflammation, we have changes in our calcium handling, mitochondrial dysfunction, and perhaps a loss of cells. And what we really want to understand is at the level of the sarcomere, what's changing in the sarcomeric proteins and the interactions that drive and allow our heart cells to contract and relax optimally. And what are the differences between men and women? So my lab focuses on using a very specialized technique where we can isolate and separate out our myofibrils. These are our series of sarcomeres, and we can take away other mechanisms of regulation. So the calcium handling that can also be perturbed with aging, the mitochondrial function that can be perturbed with aging. We can take away these factors and just focus on the kinetics of our sarcomeric proteins and how these proteins interact to get a functional response. And so if you look here, up here at this left-hand panel, you can see what we have is a double-barreled pipette under a microscope, and you can see a flow of fluid. And these are our solutions that we can control everything about, right? So now we have isolated and we've taken these sarcomeres and we can expose them to different concentrations of calcium and see how they respond. We can expose them to different ATP concentrations and see how they respond. It really gives us a nice ex vivo reductionist approach to understand how these sarcomeric proteins are interacting and what conditions in disease, sex, and age have an effect on and how it affects the function. So in this subset, this rectangle is blown up here. You can see a myofibril between our two tools, and this vertical tool is called a force probe. And this is just a micro tool that we make that we know the compliance of. So we know how much tension that myofibril can generate to move that force probe. And so when we expose this isolated myofibril to calcium in the second panel, we get a contraction event. And then when we remove it from calcium, it relaxes. And so in this, let me see if I can get out of the laser pointer here. So this is just an example movie of how a myofibril activation looks. So you can see it contracts, it moves that force probe, we push it again and we break those cross bridges and we generate, and then we can resolve relaxation. And so down here, I'm going to go back to my laser pointer. Sorry about that. You can see an example trace. So you can imagine we can get a lot of data about how these proteins are interacting and what their differences are in disease and age. And so for this project, we used isolated myofibrils from non-failing donors. So at the University of Colorado, we have access to a really great tissue bank started by Mike Bristow, where we get explanted hearts of many different diseases, as well as non-failing donors. These are hearts that can't be placed for technical reasons. And so what I did is I isolated myofibrils from male and female hearts, of different age groups. And you can appreciate here in this figure, the rate of, or the time of relaxation, duration of relaxation of the myofibrils from females over 60 years of age is significantly longer than the myofibrils from females 20 to 40 years of age. Interestingly, we also see a sex difference in our 20 to 40-year-old males in comparison with 24-year-old females. But for this talk, we're really going to focus on the interaction or the change that could be happening in these females as they're aging. So as I preluded, this talk is really focused on acetylation. And acetylation is a post-translational modification modified or regulated by histone acetyltransferases, which add this acetyl group to lysine, essentially blocking this positive charge. These lysines can be deacetylated by histone deacetylases. As the name implies, a lot of this work has been pioneered in histone modifications, but we've now found, and other groups have found, that many different proteins are acetylated, and really trying to figure out how this causes a functional changes is some of the work in my lab. And so my mentor, Dr. Mark Zhang, who's pictured here, did this study where he isolated rat myofibrils. He treated them with a P300, which is a promiscuous acetyltransferase, and found that the duration of that timing of relaxation in those myofibrils is significantly faster with an increase in acetylation. And so one of the questions I wanted to know is what's happening in these older female hearts? Is there a change in acetylation with age? Oops, sorry. So what we looked at was, by Western, we looked at general acetylation of the myofibril enriched proteins, and we found that the females of the greater 60 years of age have a decrease in acetylation compared to the younger females. And then when we did mass spec, we got several different protein targets that one of the things my lab is doing is interrogating these functionally and how they change the function of the heart. But what you can really appreciate is that in the younger females, they have significantly more acetylation than even the young, or they have a significantly more of a change with age than the young to old men. So, as I mentioned, acetylation is modified or regulated by HDACs and acetyltransferases. And you can appreciate in this picture that there are many histone acetyltransferases and many isoforms of HDACs. And really what we want to know is, are there specific targets for these distinct HDACs? And so I went back to the tissue bank and I looked at the transcriptional regulation or of our older female compared to across the age group, of all the different HDAC isoforms, and found only one was modified differentially with age in the heart tissue of the females, in particular HDAC8. So this just shows across age that HDAC8 expression is increasing in our tissue bank, non-failing donors. And then this is just another graphic divided out by age, where HDAC8 expression is upregulated in these older female hearts. We don't see this change in the male hearts. Corollarily, we also looked at HDAC8 expression in patients with restricted cardiomyopathy. These are patients that have a prolonged relaxation or diastolic dysfunction. And we saw HDAC8s upregulated both in male and female hearts, as well as in a female porcine model of heart failure with preserved ejection, which is another condition with diastolic dysfunction. So we're just starting to see a sort of a trend where HDAC8 is upregulated with prolonged relaxation. And so we went into a cell model where we can isolate the cardiomyocytes from adult rats, and then we can treat them with different things. So we infected these adult ventricular myocytes with an adenovirus overexpressing HDAC8. And we found that, as expected, total acetylation was decreased and the relaxation was prolonged. when we did the opposite, when we treat these ARVMs, or these adult ventricular myocytes, with a specific inhibitor to HDAC8, we get increased acetylation and faster relaxation. Really, and then we can also take these isolated myofibrils and incubate them with a recombinant HDAC8, again, decreasing acetylation and prolonging the relaxation. So it really leads us to this overall hypothesis with HDAC8 that as females are aging, there's an increase of HDAC8, which leads to a deacetylation and a prolonged relaxation. And this isn't happening on males. With that, I'm going to switch gears really quickly and talk about another HDAC6 and how it differentially affects the sarcomere. Really to point out that these different isoforms of HDACs have different targets, and they affect the function of the cardiomyocytes differently. So this project was done in collaboration with Dr. Tim McKenzie's group, and headed by this postdoc, Dr. Alvin Lin, who is currently a postdoc in Singapore right now. And this really started out as a phenotyping of an HDAC6 knockout. So we just knocked out HDAC6, and they were phenotypically normal until the animals were aged. So you can see here the E to A ratio is a measurement of diastolic function, and with age we have that decrease in diastolic function in normal animals, but this was more pronounced in the HDAC6 knockouts. When we looked at the myofibril function from these healthy adult animals that we didn't see a phenotype by echo, what struck us was an increase in resting tension, not in the duration of relaxation like we were expecting. And so just as a quick reminder, one of the things that we do with our myofibrils, I'm not going to pull out of the screen. So one of the things we can do with our myofibrils is we can push against it, and that can give us a reading of compliance or passive tension of that sarcomere. And so we can measure this passive factor of the myofibril. So you can imagine if we have a stiffer myofibril, our force probe is deflected more, versus a more squishy or compliant myofibril, the force probe is deflected less. And so we're able to measure reliably changes in this passive tension. And so we do this over a course of different sarcomere lengths, because one of the factors of the heart is with increasing sarcomere lengths, we get an increase in passive tension. And you can appreciate in this figure that with increasing sarcomere length, our HDAC6 knockout animals showed an increase in this passive tension or resting tension. So what happens? We don't have a phenotype in the heart at baseline in this HDAC6 knockout animal. What happens when we treat them or we put them on a model of diastolic dysfunction? And what we found was, and this model is a mouse model where the mice have a uterine injury, and they're also treated with DOCA. And so you can appreciate the wild type with the diastolic dysfunction shows this decrease in E to A ratio. But you can also appreciate that HDAC6 knockout on top of this significantly decreases diastolic function. And diastolic pressure is significantly higher in these mice lacking HDAC6 with this diastolic model. This is another way, the way I just talked about, where we increased the sarcomere length. And once again, you can appreciate the wild type animals with this diastolic dysfunction show an increase in this passive tension, but this is exacerbated in the HDAC6 knockout animals. So we once again went to our adenoviral tools, and so we can, using an adenovirus overexpressing HDAC6, we can infect the isolated primary cardiomyocytes, isolate their myofibrils, and look at their mechanics. And you can appreciate in this black line, this is our control ARVM, that's the passive stiffness from these myofibrils isolated from these cells. If we overexpress HDAC6, we get a softening or a decrease in this resting tension. If we treat or we infect the ARVMs with an HDAC6 mutant that is catalytically dead and does not cause deacetylation, we do not get a difference between our control and a change in resting tension. So resting tension is contributed to by two things in the sarcomere. The primary mechanism is titin, which is a huge structural protein, and it can also be contributed to by residual cross bridges or actin and myosin that can be interacting even without calcium. And so to figure out what was changing in this HDAC6 animals, we treated the myofibrils with BDM, and this takes away any residual actin myosin that are interacting. And then we repeated our resting tension measurements. And you can appreciate in our wild type animals treated with BDM, we do not see a change in the resting tension. Neither do we see a change in the knockouts, indicating that it's highly likely that a modification of titin is driving this change in resting tension. So titin is this huge protein. It's the biggest protein that our body makes. It's very difficult to work with. We turned to a collaborator, Dr. Hank Brazier, down in Arizona, who has a knockout animal where these animals are missing a specific region that is commonly regulated in heart failure and disease, called the PVK region. So we have these knockout mice that lack this region of titin, and we were able to treat them with recombinant HDAC6 and measure their passive stiffness. And you can appreciate, once again, if we take wild type animals that have wild type titin, we treat them with this recombinant 6, they have more compliance or less passive tension on their myofibrils. But when we took these PVK knockout animals and we treated with HDAC6, we lose this effect, indicating that while there may be other areas in titin that are modified by HDAC6, one area that could be modified is in this PVK region. So that leads to our overall conclusion that in the case of HDAC6, we have a decrease in acetylation in the PVK region and perhaps in other proteins that lead to a reduction in myofibril stiffness. And so if we have a knockout of HDAC6, we get stiffer myofibrils and we have worse diastolic function, which again goes to show that we're looking at these different HDAC isoforms and we're having completely different responses in sarcomeric function. And with that, I just want to thank my lab, which consists right now of Chris Hoffer and my new postdoc, Tim Jones, and then Tim's group, particularly Alvin, who did a lot of the work, Jen Major and Josh Travers as well, and then the rest of the team and my funding, and I can take any questions.
That's a really nice talk, thank you. Do you know, I mean, I guess the most obvious question is why is there a difference between males and females in the acetylation patterns?
I think that's a great question, and one my lab is really interested in knowing, is trying to figure out why would we have a difference? Is there a difference in the histone acetyltransferases? Is there a difference in the expression of the HDAC6 leading to this? One interesting point about HDAC8 is that it is an X-linked, or it's on the X chromosome. It's also been reported to have regulatory mechanisms on estrogen receptor. So a lot of things about HDAC8 are pointing to some sex difference. We're still teasing out. Why would we have different mechanisms to regulate sarcomere function at all between men and women?
Right. Fascinating, thank you.
Mary?
How does acetylation affect titin function? What exactly is titin being? How would it affect its function?
So titin is basically a spring unit. So if you imagine my hands are the Z discs and between them are the thick and thin filaments, titin is going to act as the sarcomere contracts, it's stretching that spring unit, and then it helps bring it back to relaxation. So it plays a role in compression and relaxation, and we do know that phosphorylation at different sites can change the stiffness of that spring. So you can imagine if you change that or you modulate the stiffness of the spring, you're pulling against a bigger load, or it's more compliant and less likely to spring back. And so all of these things have a bit of a yin and yang, like we're going to affect contraction this way and relaxation this way. So right now, from this data, we think acetylation, at least modified by HDAC6, is affecting sites that change the stiffness in a way that if we're increasing acetylation, it's becoming stiffer. It's harder to compress, and it's going to spring back faster.
I have a follow-up too, if that's okay.
Oh, sorry. No, I was just also intrigued by the female-male difference. And I wonder if you take animals, castrate them before puberty, after puberty, and do gender-affirming therapy, so to speak, would that change anything, or those male things stay in light of male participants in female sports and so on?
I mean, I think if we step back and we do the ovarectomy studies, I do not have ovarectomized data. We use a drug called VCD, and this is in collaboration with Dr. John Canelis down at the University of Arizona. We have preliminary data that we don't see in young animals that undergo menopause, because mice don't go through menopause, but if they have loss of their ovarian function and loss of their estrogen signaling, we don't see that change in prolonged relaxation that we see in the aged females. So we think that there's an age component specific, but we're still working that out, and that's one of our key questions is how and what's happening in the males, and we haven't even touched that yet.
No, but those oophorectomized animals, you didn't give them testosterone and see whether that.
Oh no, and I mean I think those studies we have a long ways to go.
Okay.
We've got a model in rats, not in mice. Can you do these experiments in rats? Or
I can do this in a lot of things, yes.
Because we have hearts from rats that have undergone ovarectomy with either estrogen or estrogen plus FSH add back. Yes. So it's a 2x2 model. So they might be of interest in terms of.
I would like to add different hormones.
Yeah, I know you want to add different. I know. but
and in males as well.
Yes.
And do it before they reach sexual maturity, and then once after sexual maturity.
I mean, I think we have so many questions, and I'll reach out. Thank you.
She has all this animals.
Can you work with her with smooth muscle?
Not smooth muscle. We have, but Dr. Walker is an expert in the mechanics of smooth muscle. We can do skeletal muscle. We can also do many different cell models, so iPS cells, as long as the sarcomeres are mature. With that, I will say thank you.